Go ahead and get started today. It is my distinct pleasure to introduce Dr. Bob Sterner to give our seminar today. Bob is coming to us from the University of Minnesota-Duluth. They are one of the latest partners of the Seiler Cooperative Institute around the Great Lakes. So Bob graduated with his Bachelor's of Science in Biology from the University of Illinois, and then ultimately his Ph.D. in Ecology from the University of Minnesota. Ever since that time, he has been interested in linking biological and chemical approaches to understanding lake ecosystems, and he's probably best known as being one of the founding fathers of the field of ecological stochiometry, which is one of the few subdisciplines in ecology that truly links the physically based ecosystem dynamics to the biologically based community ecology. Bob has focused mainly on freshwater plankton to test his ideas throughout his career. But through his interactions with other colleagues, he's worked on everything from microbes to, I think, elephants as well. So at least in principle with his models. He's published an insane number of papers, approaching 100 that have been cited an insane number of times by his colleagues, and he's won all sorts of professional awards for his work. But I'll just highlight one. He was honored this year with the John H. Martin Award from the American Society for Limnology and Oceanography for a foundational paper that represents a paradigm shift in limnology. And that was for his, 1994 paper written with Dag Hessen. It was in Annual Reviews, and it was called Algal Nutrient Limitation and the Nutrition of Aquatic Herbivores, one of his early sort of seminal works on ecological stoichiometry. After he graduated with his Ph.D., he had his first faculty position at the University of Texas, Arlington. He then moved in 1994 to the University of Minnesota at the Twin Cities, where he ultimately became head of the Department of Ecology, Evolution, and Behavior. And then he took a 2-year hiatus from 2007 to 2009 to go be the director of the Division of Environmental Biology at NSF, where he managed about a third of the nation's portfolio in environmental research, over $120 million a year. And he since, very recently, in 2014, moved to the Great Lakes Observatory, the Large Lakes Observatory, up at Minnesota-Duluth. And I think he's going to talk now about some of his work that's been going on up there. So help me in welcoming Bob Sterner.
Hi, everybody. Thanks, Brad, for that great introduction. It's a pleasure to be here. And is the microphone okay? We're online and everything. A lot of people online. Hello to all of you online. Welcome to the talk today. And so Brad's introduction emphasized the work that I've done in kind of joining chemical limnology and biological limnology. Today's talk won't have very much of that in it. Today's talk is more about the way what we know about this really big lake upstream of you here. And how it's changing. So we're going to be talking about some of the basic limnology of Lake Superior today. And the title of the talk is Lake Superior, a Warming Ecosystem, because I'm going to try to convince you that climate change is a principal driver in the lake, and then also try to show you what we know today about how climate change is affecting the lake ecosystem. I want to mention Kate Reinl, who's here in the audience. She's my new Ph.D. student. And she was instrumental in a part of the talk today, so we'll get to that in a little while. And part of the data that is used in that part of the talk comes from Jay Austin at University of Minnesota-Duluth. So, this was working before. That's all right, I'll stay up here. Okay, I'm frozen. Well, the arrows don't work, but the mouse does. Okay, great. Thank you. All right, so let's start here. This is not my work. This is from the O'Reilly et al. Paper that was very well cited about temperature trends in lakes around the world, and many, many lakes around the world are warming. And this figure actually was produced by John Lenters, who I guess isn't around today. I don't see him out there. But he put this together for a proposal the two of us were working on. And the point of this slide is that although many lakes around the world are warming, They're warming at different rates. And if you just think about summer surface temperature, not total heat content, but summer surface temperature, Lake Superior is one of the fastest warming lakes on the planet, which by itself is kind of a staggering thought, this gigantic inertial mass of water in Lake Superior. How can it be warming so fast? Well, we'll talk about that in a little while. But this graph shows the box plot of warming lakes and E and C on the, I'm going to point with this so that people online can see it. E and C are the eastern and central buoys of Lake Superior up here in the upper tail of the distribution. So there are a few lakes that are where temperature increases have been measured to be even more rapid than Lake Superior, but it's one of the most rapidly warming lakes on Earth. What can this be doing to lakes? This is something many of us need to be concerned about. We need to have a good science basis to understand how climate change is affecting lake ecosystems. And the truth is, like anything else in biology or ecosystem ecology, it's complicated. If we warm the landscape, many potential things can happen. I pulled this list out of a paper by Adrian et al. From Limnology and Oceanography in '09, and the point is just there's lots of things that can happen to a warming lake involving hydrology, the temperature of the lake water as we've described, thermocline depth, all these things can happen, and other things that aren't even mentioned that I added to the list, light delivery, the sort of seasonality of the cloudiness or the amount of light hitting the lake. Wind is probably a huge factor that we don't have a good handle on. Storminess. So it's a rapidly warming system, but getting beyond that is a very difficult question. What does that mean? Is it, quote, good or bad, let's say, for fish populations? There's a lot of things we don't understand about that, and today is about trying to kind of get some foothold on that question. So if you read the literature on this, you'll find that there is a sort of a cluster of papers that are talking about a similar phenomenon in warming water columns. There's the work in Lake Tanganyika by Verberg, by O'Reilly. A couple of other people have followed up on this. And it basically says, it's a little controversial, but it basically says that one of the principal things that happens to a warming lake or a water column is the higher temperatures of the epilimnion mean that you have an increased density gradient between upper and lower waters, and that higher density gradient reduces mixing of nutrients from below to the euphotic zone, and therefore a warmer lake, all else being equal, should exhibit lower production and declining fisheries. And again, a little bit of a controversial example with Lake Tanganyika, but that's the mechanism that's been proposed there. The exact same mechanism with appropriate modifications for the ocean has been advanced for thinking about warming oceans. So there's a handful of papers, this Barenfeld et al. Nature paper is one of the main ones, that talks about the same thing. A warming ocean cuts the delivery of nutrients off from the bottom to the top or reduces it, and therefore results in declining production. So this is one of the main hypotheses out there about what should happen to warming water columns, is we should see lower nutrient delivery and lower production. So what I want to do now is take you to Lake Superior, the headwaters of the Great Lakes, 10% of Earth's fresh water, a place that I've been studying for many, many years through a series of projects. And through all of those projects, no matter who funded them, we attempted to make a single set of core measurements, often at the same place in the lake, or at least similar places in the lake, to try to stitch together a time series. Even though we didn't have long-term funding, we had project-by-project funding, and over the years we've attempted to create something like a time series from that data. It's not perfect in terms of time series, but it's all we've got. So we're going to approach this default hypothesis, quote unquote, basically saying this rapidly warming Lake Superior should be exhibiting declining production, et cetera. The other point to make is, though Lake Superior is one of the most rapidly warming lakes on Earth, other anthropogenic forcing factors are relatively muted. So there are invasive species in Lake Superior, but the story there is nothing like Lake Michigan, where the entire ecosystem has been converted from one set of conditions to another. There is urban runoff into Lake Superior, but it's a trickle. It's nothing. There's basically no agriculture in the watershed. 95% of the watershed is forested. So these things that we usually think of in terms of major anthropogenic forcings, they're there, but I argue, and I think it's a defensible point, that if we want to understand climate change, we want to go to a place where climate change is big and important and other things aren't. And I think Lake Superior is an example of a place like that, which makes it really valuable for us to say, what are the effects simply of climate on lake ecosystems? So some observed trends, these are things that have been published. I'm not sure if any of the authors of any of these studies are in the room, and they may be. Remote sensing approaches have been utilized to try to go back in time and try to observe whether there's been a change in the optical properties recorded from space. And two papers here, one from JGLR 2012, one from JGLR 2017. Barbiero, first author of one paper, and Yusuf, the first author of the other paper, basically saying the same thing. From a remote sensing standpoint, there doesn't seem to have been an observed change in the transparency of the lake as inferred by remote sensing. So in this case, with the green, we have two different years, 1999 and 2006, over a seasonal cycle with very little difference between the 2 years. And here are two different measurements from two different satellites for Lake Superior with basically no observable trend over time. So it would seem that though the lake is warming, these data say that's having no effect on at least the turbidity of the water. Paleo Studies are emerging. Ewan Reevey and his colleagues at the Natural Resources Research Institute are doing a fantastic job on some detailed stuff about algal community structure. And one of their recent papers in Limnology and Oceanography identifies one particular type of diatom, a cyclotella, that is becoming increasingly common actually throughout the Great Lakes. And in this paper, they argue for a very strong connection between the increase in this cyclotella and climate change. It is a paleo study, so it's not a direct test of climate per se, but it's making an argument that something about climate change is affecting the algal community and in fact increasing cyclotella. So there are others that I could do, but this isn't really a review paper, it's a research seminar. Work that has gone on over my projects that I've shared with many different collaborators is showing actually a very different picture from these things that I've just mentioned. So this is directly measured chlorophyll concentration, algae caught on filters at a single sampling station in the western arm of Lake Superior. We call the station CD1. And again, over the various projects, we've gone to this station and done basically the same thing over time, measured chlorophyll in the water at a variety of depths. And I've simply plotted all of the surface chlorophyll data that we've collected at this one site. So this is observations of chlorophyll made in 20 meters or less depth. With the earliest data, which come from the 1990s in red, and then later periods of data, blue, black, and this sort of brownish color. And it's very clear, I think, from this that the lake has changed. In fact, algal abundance has dramatically dropped. To remind you, this is just surface algae plotted here, and it's dropped over a relatively narrow window of time. So it's potentially still consistent, say, with the remote sensing observations, because we're just looking at a segment of the algal community here, the surface algae in a very specific part of the year. But there seems to be a really big difference between the earliest observations and more modern ones. Here's similar depth in the lake. This was less than or equal to 20 meters. I haven't updated all these graphs, so it gets a little apples to oranges here. But this is now that same segment of the year, Julian day 190 to 255. These are all these chlorophyll observations we made throughout the water column. Again, red is the earliest, then blue, then black. And you see basically throughout this reduction in chlorophyll. This is starting to look quite different from what, say, the remote sensing data have to show us. Now, this is one place in the lake, but it is directly measured chlorophyll. So, you know, different data sources may be more or less convincing to you, but there seems to be some kind of a contradiction here. In addition, it seems to be especially the large algae that are dropping in abundance. This graph really interests me a lot. So this is annual means of chlorophyll directly measured in different size fractions, less than 80 microns, less than 10 microns, less than 3, less than 2. Again, things differed a little bit project by project by what we were doing. But if you'll look at the black dots, the less than 80, chlorophyll dropping very significantly, again, surface conditions, et cetera. But the less than 80 fractions dropping at a rate of about 0.7 per year. The next fraction down dropping at a rate of about 0.1 per year, and the pico fraction basically dropping but not so fast. And so I could throw many graphs like this at you. The trends are backed up by measurements of particulate organic carbon. So it's not just pigments, but it's actually particulate carbon in the water. We can slice and dice the data in many, many ways. If you'll just take my word for it today, the strongest signal that we're seeing is it's the large summer surface algae seem to be dropping out of the system. So we're kind of left with somewhat contradictory messages about what's happening in the lake. No change in transparency. The Reeve group showing one particular type of diatom increasing. These chlorophyll data indicating a drop. This is kind of a sad state of affairs for Great Lakes science in that we don't have a single place to go to get the definitive data to tell us what the trajectories of this ecosystem is. And at the very end of my talk, I'm going to come back to that point and say we need to do a better job with just the routine monitoring of these systems than we're doing now. But that's where we're at. I would say, personally, you know, my data convinces me most strongly, but in reality, the literature has somewhat contradictory messages about what's going on in the lake. So we're going to, now for the rest of the talk, we're going to take a different approach. Again, we're going to study the lake at different years and different seasons, mostly in the western arm of the lake, but we're going to evaluate the conditions of the lake that we measure and express them all relative to surface temperature. So surface temperature is seasonal, obviously cold all winter, warms up to different degrees in different years in the summer, but each year gives us some variation in surface temperature, and we're going to take data and just see how much of the variance in lower food web properties we can explain with surface temperature alone, as if that was a warming lake. The first set of data is, I believe, the only offshore primary production data measured over time in any of the Great Lakes. If I'm wrong about that, please correct me. But over these projects that I've described, I have been involved in measuring primary production using very traditional C14 technique, in situ incubations, all at this red dot location here in Lake Superior. So we have a temporal record of primary production in the lake, not long enough year to year to year to really say what's happening over those years in primary production, because those measurements are all made at different seasons. It's a little difficult just to plot things versus year, but we have very different surface temperature conditions where we've measured this at. So when I set this up, I purposely wanted to use a very well-established technique, for better or for worse, but data that we could compare to other data. So we used the Goffs Protocol from the oceanographic community. And this is very classical stuff. You collect water out of the lake, you bring it on board. You collect water at night, you bring it on board, you spike it with C14 bicarbonate, You put it back in bottles, incubate it for an entire daylight period, reharvest those bottles the following nighttime and filter them and see how much radioactivity is on the filters. It's usually stated that this technique measures something between gross and net primary production. But there's a lot of literature about this, and exactly what you're measuring is argued about. But again, it's a technique where things can be compared from site to site. So here are all the different primary production measurements we've actually made at that one location, starting in 2006 and ending in September of last year. We don't plan to do any more this year, so it's kind of a good time for me to just take a look at the data and see what it says. These are the different projects that have been involved, funding sources, and the various collaborators who have worked with me on these different projects. So we've measured in situ primary production in this gigantic lake a total of 21 times. And you can just kind of scan down the months, the leftmost numbers in these dates, and you can see we've observations in November, we have observations early in the year. Some years we did a lot, some years none or a few. But this is what we've got, 21 of these measurements. This is just a fun picture. This is the boat we work with, the Blue Heron. I'll tell you a little bit more about the Blue Heron at the end of the seminar. And the big collection of floats here has a cable dangling from it with the bottles basically incubating in the lake. And then there's a little drifter next to it to help us relocate this free drifting array of bottles at the end of the day. So it's a radar reflector and a strobe light on it. Okay, so just some example profiles. This won't shock anybody in the room. Primary production's higher, more or less, near the surface. It drops off where the lake gets dark. Sometimes we see these surface inhibitions, not always. But this is just a collection of profiles to give you a feel for what the data are here. I find this particular result quite fascinating. So this is primary production in Lake Superior at the bottle scale. So every dot on this three-dimensional graph is a bottle that was deployed in the lake with primary production as measured by C14 and the vertical axis, and light and temperature are the horizontal axes. And so it's probably hard to see, but this is the light axis here, and this is the temperature axis here. And you can see where light's low. It doesn't matter what temperature it is. Algae aren't going to grow. It's always dark. And the light response is visible through this series of hyperbolic relationships accelerating with temperature. So this 3D relationship gives us both light and temperature information. The most interesting thing to me about this graph is those two predictor variables, light and temperature, explain 87% of the variance in primary production in Lake Superior. We don't even have to tell the model how green the water is, how much chlorophyll there is. This is just the rate of carbon incorporation. There is no biology in here. This is all physics driving primary production, which is bad news for me as a biologist. But in some ways it's good news because it suggests a level of predictability of the system that we can really use for lots of things. So if it's truly the case, all we need to know is light and temperature, we can model this whole ecosystem in a really robust way. For today's talk, we're going to collapse this data and look at it in the simplest way possible. We're going to take those 21 observations made over those many years and just calculate the integral production under a unit surface of the lake. So in this particular deployment, it's 188 milligrams carbon per square meter per day. So we basically have 21 of these now, and we're going to try to see if they'll tell us anything about what happens in a warming lake. So this is the frequency distribution of measured primary production integral, and it ranges a lot from something like 100-ish milligrams carbon to almost 5 times that. And there seems to be this one bar here that's way bigger than the others. There's some condition that we have frequently observed with enough skew in the data that the mean doesn't do a great job of describing the most frequent condition, but that's not the most interesting way to look at the data. It's just the frequency distribution of observations. This is now taking those 21 observations of primary production and plotting them against the photon dose hitting the top of the lake for that day. So this is simply asking, does light alone control aerial or integral production in Lake Superior? And the answer is sort of, but not much. And the sort of part is the 2 times we have done this in November, and I pause for a moment to have you think about us working in Lake Superior in November with radioactivity on board. Those are interesting times. But those 2 observations we've done in November, low light, low production, so there seems to be something going on, just seasonal light supply, but otherwise light doesn't do much. This is probably the most important graph for this part of this talk. This is, again, just looking at integral production, now simply plotting it against surface temperature. And remember, the default hypothesis for water columns is that if they're warmer, we'll observe lower production because of reduced nutrient input. And these data are very, very hard on that hypothesis. There's a very highly significant positive relationship between primary production in the lake and surface temperature. It's also the case, now this is, I'm pushing the data probably a little further than they deserve, but I am interpreting these data presently as 2 different states. The state below 8 degrees surface temperature and the state above 8 degrees surface temperature. So below 8 degrees, the data are pretty variable, and the mean is 232. And above 8 degrees, the data are much less variable, and the mean is 364. So again, I admit we're pushing the data kind of hard here, but I would say that what we're seeing here is a warming lake means more carbon going into that ecosystem, but at that point above 8 degrees, if we just looked at the data above 8 degrees, we would see no statistical relationship with temperature. So whether the lake is 10 degrees or 15 degrees or 20 degrees doesn't seem to matter a whole lot. It's just in its warm state. And so what does that mean? Does that possibly mean, well, Lake Superior may be one of the fastest warming lakes on Earth, but the base of the ecosystem doesn't care. It's operating in that warm lake state. And I would say, before we get to that, the light variable does not go away. In fact, it becomes highly significant when we correct for temperature. So in this graph, all I've done is looked at the residuals of the data versus temperature and plotted those residuals against light. And now things are sensible. So once we correct for temperature, now we see a highly significant positive relationship with light. Again, light and temperature are strongly driving this ecosystem. Okay. So above 8 degrees, there doesn't seem to be much going on. Does that mean we don't have to worry about climate change? And the answer to me is not so fast. The reason is a warming lake isn't just reaching a warmer temperature, but it's stratified for a much longer period of time. And so this, again, a figure courtesy of John Lenters, but this is the summer onset of stratification in Lake Superior, with warm lakes, warm years, 1988 and 2012, the lake stratifying in very early June. And in cold years following a lot of winter ice cover, the lake stratifying not till early August. So there's a very long gap between an early and a late stratification in Lake Superior. This is mooring data taken from Lake Superior, contrasting cold years and warm years. Really interesting stuff going on here. So we come out of the winter, the lake is always cold in May, down here with about a 2-degree difference in temperature in May, and the lake warms at about the same rate in a cold and a warm year. The red here has about the same slope as the blue. But the point at which surface temperature reaches 4 is critical. Once the surface temperature reaches 4, incoming heat is concentrated within the epilimnion. And so the rate of warming of the surface goes way up as soon as the lake hits 4 degrees. So the reason we have this huge difference in stratification date is that's the difference at which the lake reaches 4 degrees. And in a warm year, we follow a trajectory like this. And in a cold year, we follow a trajectory like the blue. Basically, in a warm year, the lake certainly does get much warmer, but it's stratified for a much longer period of time than in a cold year. So we can put all this information together. Oh, there's a cold, the stratified season in a cold year and the stratified season in a warm year. So we can put all this together and just say, okay, that 8-degree breakdown, that's an important one. If we contrast a cold and a warm year, how much carbon is going into this ecosystem under these different conditions. And this shows in a cold year how much primary production there should be between June and October versus a lake that stratifies for 30 days versus a lake that stratifies for 100 days versus a lake that doesn't exist but which was stratified for that whole period of time. So there's this accentuation. The amount of new biomass being created in this lake varies a lot between a cold and a warm year, not so much because of a temperature-dependent response per se, but because of the length of the stratified season. It's that interaction between the physical habitat and biological response. It's really important here. So biology alone isn't helping us a whole lot. Physics alone isn't telling us anything about the ecosystem, but it's that intersection between physics and biology that I think we need to pay attention to. Okay, so the next part of the talk is the part that I got assistance with Kate, and she's been working hard on this data all semester, and some of the data in this part of the talk comes from glider data collected by Jay Austin and his group. So we approached the data that we had available to us with the question, would climate change affect the vertical structure of the algae in the water column? The algae aren't perfectly mixed in the water column. There is a always present deep chlorophyll maximum in Lake Superior. There's an important vertical gradient in this lake in terms of primary producers. What can we say about what climate change may do to that vertical gradient? And so to do this, we worked with CTD data and bottle chemistry data taken between 2014 2016, and then glider data taken from 2011 to 2015. For this part of the talk, we only looked at when the lake was positively stratified, so essentially only the warmer parts of the year. We only looked at locations with depths greater than 75 meters. We looked at nighttime data only because the in vivo fluorescence measure is more reliable there. And in the case of the glider, which gives this exquisite spatially resolved picture, we took each transect of the lake from one shore to the other and made one data point out of it, basically averaged an entire transect just to get an observation of a transect over this period of time. But doing this, we had a lot of spatial data on the vertical structure of the phytoplankton community. So one of the things we spent a fair bit of time doing was asking ourselves, can we trust these in vivo fluorescence measurements to tell us anything sensible about the deep chlorophyll maximum? Because you can run a fluorometer up and down through the water column, but there are many reasons why those in vivo measurements may depart from actual chlorophyll in the water. And what we came to decide, and again, I could show you many slides in this, but I won't. We came to decide that the in vivo measurements are perfectly good at telling us about the position of the DCM in the water and its overall shape. But we didn't want to rely on it for absolute measurements of how much chlorophyll was in the water column. So absolute measurements, we relied only on the extracted values. Size and shape and position of the DCM, we use these in vivo measurements where there's really good spatial information. The bottom graph is a graph of in vivo measured versus extracted chlorophyll, many, many data points here. And they're scattered, but it's unbiased. The one-to-one line is a pretty good measure of the central tendency. So To describe the DCM, we have many measures of that as well. I'll be showing you things about the depth of the DCM, the depth of the peak of chlorophyll, and also what we call the full width half maximum, essentially the measure of how narrow or wide the DCM is. Before showing you the things where we found some good relationships, I think this is an important piece of the interpretation here. It turns out that thermocline depth, which we also had estimates of, is not very well related to surface temperature. And I show this because I think nearly every conversation I've ever been in about this topic in general, in that conversation somewhere, the person I'm talking to will automatically jump in and say, well, obviously in a warmer lake, the thermocline's deeper. Or the next person I'm having a conversation with will say, obviously in a warmer lake, the thermocline will be shallower. And it turns out it's just not a good relationship, period, in this data. There is no good relationship between thermocline depth and lake surface temperature. So that's an interpretive piece that we'll use to understand the rest of the data. These relationships are noisy, statistically significant, and positive, but these are measures of the total algal abundance in the water column versus surface temperature. And so in the left, we have the integral fluorescence from in vivo measurements, which we're a little spooked about using the absolute values, but there they are. There's lots of data. And the right is fewer data, but it's the actual extracted values. I'm not going to try to suggest that these are good statistical fits in any way, but the important thing here is the data here are pretty tough on the default hypothesis that a warmer water column is lower production because of lower nutrients. The evidence here is that warmer water columns have overall higher algal biomass, not lower. So if this is a climate change story, a warming lake should give us more algae, not less. This is a fascinating plot that we're still working on interpreting, but this is the depth of the DCM versus surface temperature. And in the cooler part of this data, below about 12 or 13 degrees, highly, highly variable. You get up above, say, 13 degrees, and the variability drops, which means the mean depth is shallower at warmer temperature, but probably the story here is more about variance than mean. The fascinating thing to me here is the big differences from year to year. So the different colors are different years of observation. And I'm not really sure where to go with this, but it's very clear that the different years are doing different things in this depth of DCM versus temperature. And I'm going to leave that essentially as a mystery right now. We don't have a good explanation here for what's going on. But overall, as a collection of data points, warmer conditions on average, with a lot of variance on average, are shallower DCMs. This is one of the best statistical fits we were able to get. This is the width of the DCM plotted against surface temperature with a really strongly declining function here. So warmer surface temperatures mean narrower DCMs. So if we put all that together, this is just a schematic, but this is the way we're thinking about the vertical structure of this community under different temperature conditions. Blue, of course, is cooler, red, warmer. Bluer conditions, we have lower overall algal abundance and a deeper and wider DCM. As the lake warms, the bulk of the algae move up in the water column. A warmer lake means shallower algae. And so this is kind of where we're at with these data right now. If we think about this warming condition, should we be thinking so much about the fact that it's warmer per se and things are faster when they're warmer, or that the warming condition restructures the community, pushes the algae up, where again it's really physics controlling the biological response. And I'm really interested in digging further in the data to try to see how far we can go with this. So warmer, shallower, brighter. might be the best way to think about this, rather than warmer, warmer, warmer. So those are the parts of the talk, and I am going to admit there's some contradictory information in here. Exactly what's happening to this ecosystem over time is not entirely clear to me. I think we have some trends that I personally trust, but they don't line up with what other people are saying from other kinds of data. The fact that we don't even know what the ecosystem's doing in time is a scandal. We lack the basic information about what the bulk of this ecosystem is doing in the offshore from year to year to year. I would argue that we can not even parameterize a believable ecosystem model, I'm going to say, for any of the Great Lakes right now. The gaps in knowledge are just mind-boggling. The fact that we're struggling even to understand what, first order relationships are of primary production and temperature in these systems. If we're so, if our knowledge base is so weak where that's still, sort of the question that we have to deal with, how can we potentially think about these systems going into the future? I think, and one of the things I'm interested in being here in Ann Arbor to talk about is we need to think really hard about how we're observing these systems. And I would argue for a community effort to try to talk about what the important gaps are in terms of understanding these systems. Different groups doing different pieces of this, but I think we should step back and say, what do we really need to know to understand the long-term trajectory and why these systems are changing the way they are? So I think that the evidence to date is very hard on that nutrient hypothesis for Tanganyika, for the oceans. I'm pretty strongly thinking that a warmer lake means more production, but again, we have some contradictory information and some struggles to understand how to line data up. I want to close with a few more or less announcements. Almost all the data I've shown you, not quite everything, but the background carbon chlorophyll data for going back to the early 2000s and up to the present, all of those data are publicly available at this website, BECODEMO, which is a data management organization funded by NSF. We have uploaded all of that background data there. If you want to see what's going on Lake Superior, I invite you to go to that website under the program Laurentian Great Lakes Ecosystem Studies and download the data and look at it to your heart's content. I want to announce a summer undergraduate position available in my lab. If you are interested or know somebody who might be interested in coming to Duluth for the summer, we're looking for somebody to help us out with our nutrient addition studies this year. I mentioned the Blue Heron. This is our research vessel. It's the only UNOLS vessel on the Great Lakes. It's available for work in the Great Lakes. It can work 24 hours a day. We're always interested in having more people use the boat. Talk to me or write to Doug Ricketts at the email that's at the bottom of the slide to make use of the research vessel. It can go anywhere in the Great Lakes. And the last thing, I do hope there would be some interest in this. There still is some time to apply for the chief scientist training cruise that's coming up this year. This will be the 4th year that we've done this, but basically we take applications from people from really all over the country, and they get on board the Blue Heron, they get some ship time, they work as a team to make sampling schedules, they get some preliminary data, they get a chance to see what working on an oceanographic vessel's like. We've had people from the oceanographic community, we've had people from, I don't know, is it saying here how many? 25 participants from 15 institutions so far, and applications are, the deadline's coming right up. But if you are interested or know of somebody in your lab who might benefit from an experience like this, to learn how to do this kind of work, but also to get out on the lake, basically for free. Their stipends are paid, their travel's paid. It's a great experience. There's still time to apply for this, so I hope we'll see some applications come from Ann Arbor. So that's it. Thanks to funding agencies, all the collaborators, crew of the Blue Heron, and to Sandy Brovold, my secret weapon lab manager who helped collect a lot of this data. That's it. Thank you.
We have some time for questions, but I'm going to pass around the microphone so that the people who are online can hear the questions before Bob answers them. Anybody?
I'm trying to wrap my head around how the default hypothesis works in Lake Tanganyika,
Yeah.
and how that compares to Lake Superior, where the stratification is much more seasonally dependent, and and how that affects your whole series of results here.
That was great point, and I think so. To me, what was sort of implicit in your question is, why would Lake Superior, which does holomixis twice a year, why would we even compare it to something like Tanganyika or the oceans, which are permanently stratified? Fair point. And maybe that is the main reason why Superior is different from these systems. But the interesting thing then is what that says about how the lake works. So if it's true that this twice a year resetting and connection of bottom to top means the hypothesis, for example, for Tanganyika does not apply, I'll buy that. But that suggests that the course of the lake is set in the beginning of the year when there's holomixis. And what we see throughout the rest of the summer was basically those conditions were set when the lake was mixing. That is, during the summer when the lake's stratified, the mixing of nutrients from bottom to top is not important. And that may be true, but I think that gives us new insight into how the lake is working. So do we just start out with a unit of nutrients in the spring and the ecosystem just keeps reworking that through the summer? Or do we have to consider movement of nutrients from bottom to top? And so I think you're right that that's maybe the first place to look to understand these differences. But I don't think that's boring. I think that's actually interesting. It's providing insight into how the lake works.
Thanks. That was a really interesting presentation. I was interested in, so in the beginning you showed your plot of primary production as a function of light and temperature, and you said it was amazing that it could explain 87% of the variance. But then at the end you said it was so complicated that we can't begin to parameterize an ecosystem model.
Right.
So it's kind of like two different statements as to it's so simple that it's straightforward. And that part, I mean, was consistent with your final conclusion, which was warmer lake means greater production, and that's consistent with some of the previous primary production work like Van and Steele et al. And others, and the relationships from the ocean and stuff that just, as it gets warmer, higher productivity. So I was just interested in those simple versus complex.
Thank you, because I see what you mean. That sounds like a contradiction. I don't think it is. So the simple part, the part that was all physics, was what's controlling the input of carbon to a unit volume of the lake. 87% of that is light and temperature. That part, I think, is very simple. That's only one step in the functioning of the food web or this ecosystem. For example, ultimately, we'd like to know what food the fish have available to them. And so the complicated part comes into understanding the fate of that carbon. And that's where I think we don't have good parameterized models that have a lot of biology in them about how these systems work. So I do think it's good news that parts of this can be reduced to some very simple explanatory variables, but that doesn't mean we have a grasp on the ultimate appearance of biomass at various trophic levels and how energy and nutrients are moving from organism to organism. That's the complicated part.
We have a couple of questions online. I'm going to hit those real quick. The first one is, even in warm years, Lake Superior is still pretty cold compared to, say, Lake Tanganyika. Do you think that as Superior continues to warm beyond its current warm, it will enter a new state of productivity?
One can only guess about that question. It's a fun question. I appreciate it. I mean, to be a very conservative, don't go beyond the data scientist, we saw no hint of that in the studies we've done. We don't see any hint of some new transition happening at the really warm state. But there are other things that could be happening that we have to pay attention to. Climate change means increased storminess, for example. And what would it mean to have a so-called 100-year storm event every year or few years with the inputs that those bring? So I don't feel like I can provide a kind of robust scientific prediction about whether we can experience a new state or not. I think if we continue to collect data, we'll be in better shape to make that prediction. But right now, it's really guesswork.
Thank you. One more is, I am curious on the impact of shallower algae on carbon cycle in the ecosystem. Do you think this change will change the status of carbon cycle in the lake?
Cool question. So shallow algae, there is benthic production. Obviously, there is benthic production in basically every lake. These nearshore zones are really important for fish habitat. They're reservoirs of biodiversity. My bias is that the lake as a whole is just so dang large that the edges are relatively small in importance compared to what's happening in the water column. Stable isotope data collected offshore, I think, backs that conclusion up. So nearshore, localized specifically for populations that many people care about, nearshore fishes, yeah, it can be super, super important. Lake-wide, I think if you get the water column right, you've really gone a long way to understanding the lake.
Bob, I have a quick question for you. We got a couple more. Sorry to interrupt. I'm trying to make sense of two different parts of the talk. The first one that's showing you're losing large size fractions of algae. And I'm wondering if you have any idea why the large size fractions might be most sensitive. But if you're losing the large fractions, then yet productivity is not necessarily changing or not dramatically. It suggests there's some turnover and compensation by the smaller things.
Yeah.
So what's driving the shift? So,
you're wondering the same thing I am now with the contradiction of the data. But what we now understand through the work of Kate's on the DCM is the observations which I showed you were all for the surface layer. But I think to really understand the ecology of the system, we need to do a better job with that vertical gradient. And for example, the observation of Unrevi and others that there's a lot more cyclotella to be found, it may be the case, this is pure speculation, but those shallower DCMs that are a little brighter, maybe a little warmer, those may be perfect cyclotella habitats. So there are some apparent contradictions currently in this data, and I think we're going to have to work to resolve those because all of this is true at some level or another.
This is related to the first question you got on the webinar, but have you looked at the Tanganyika data and the Superior data on the same plot? I'm just wondering, I'm thinking the Superior data, the variance in any dimension may be small relative to Tanganyika, such that they may be more or less consistent with one another, but you can't pick up much when you're just looking at Superior alone.
Yeah, that's a really cool, the answer is no, I haven't looked at that, and I think I will now that you've suggested it. That sounds like a really cool idea. It's a little apples and oranges, so we've made one kind of measurement here, and so one would have to do that carefully, but yeah, you know, in a way, maybe Lake Tanganyika is just the ultimate extrapolation of my little exercise in how long the season is, right? So maybe that's a good endpoint for, you know, thinking about how deep water columns work. It's worth a look.
Time for one more question.
You mentioned the concept that maybe the summer season is set early in the spring when mixing happens. and that vertical transport within the season is a bit of an unknown. Would one way to approach that be to look more intensively at upwelling when and where it happens?
Yeah, for sure. Good data on mixing and upwelling is critical, coupled with good nutrient analyses over the water column, because you can have all the mixing in the world, but if the nutrient concentration gradient's small, it's still not bringing many nutrients. And the sad fact about Lake Superior is we are still, I talked about big gaps in knowledge. The vertical structure of phosphorus is a huge gap in knowledge about this lake because phosphorus is at low enough concentration that the routine measurements basically all say, yes, there is phosphorus in the water. But they don't resolve the signal against the noise very well. And so that's been an effort in my lab for some time, is to improve these, the precision of these phosphorus measurements. So we could say, what really are these vertical gradients like? Currently, I'm of a mind that upwelling probably is not a significant thing in the lake because no matter what the mixing is, we don't see conditions of high phosphorus below the thermocline. But we're looking at very noisy data, and I'm holding back until we improve our methodology to try to get a much better signal out of that data. But those are pieces of that. And it fits in with the idea of, well, maybe, you know, it was a straw man argument on my part to put up oceans in Tanganyika because A, the place like Superior mixes twice a year, and B, those vertical gradients aren't maybe very significant. So that could be teaching us why the system's operating differently. Sure.
Well, I know we have some more questions, and I'm sure Bob will be happy to stay up here afterwards, but we have run out of time. But join me one more time in thanking Bob for coming and giving the talk here.